Hello students, so in my earlier video I have explained to you about the meristematic tissue in plants. So today we'll be discussing the other type of tissue found in plants, that is permanent tissue. So both simple and complex permanent tissue we'll be discussing here. So when we talk about permanent tissue, the cells of permanent tissue are those which do not divide any further and they differentiate. They'll change their form and they will assume some special function. As we got in meristematic tissue, the cells were in a state of continuous division. They will divide, keep on dividing, and the plants will grow in size. And the moment they finish dividing, they will stop their division, they will modify themselves, and they will start some special function, and then they are called permanent tissue. So simple, now why some are called simple and some are called complex? It is depending on their composition. Some permanent tissue are there which are made up of the same type of cells throughout, and complex permanent tissues are those which are made up of different type of cells. We'll see that. So that was the definition of simple permanent tissue, which are permanent tissue of plants which no longer divide and they are made up of the same type of cell. They're basically of three types: Parenchyma, Collenchyma, and Sclerenchyma. Now this is the cross section of the cells. Now what do you mean by cross sections? There are two terms you need to understand in biology. Suppose there is a stem. If I cut a stem longitudinally, the kind of view that I will get, that is called LS, or longitudinal section. Now if I take the stem like this and I slice open it like this, I'll see this view. So this particular view, it is called the cross section of the plant. So this simple permanent tissue, these are all cross sectional view of the three types of tissue: Parenchyma, Collenchyma, and Sclerenchyma. So there are different properties based on which we can write the difference between them. First is the shape. So when we talk about the shape of parenchyma cells are isodiametric. Now what do you mean by isodiametric? Isodiametric means something which is spherical in nature. Suppose if you take a ball, from all sides the diameter will be same. Iso means same and diametric. Suppose you take a stone, a round stone, approximately it will have the same diameter throughout. So these cells are like that. Another way to understand, you have seen thermocol. So the small beads inside the thermocol, you have played by breaking thermocol. So the small beads that comes out from inside the thermocol. So this tissue is like that. So if you take a longitudinal section or a cross section, it will appear like that in appearance. Collenchyma, these are elongated cells, but in a cross section they appear to be polygonal in their outline. So the cells are elongated like this, but in a cross section they appear to be polygonal in cross section. Sclerenchyma, they are in the longitudinal section, they are long spindle shaped cells, but if you take the cross section, you'll find that the cells are actually hexagonal in shape. Now the nature of the cells, Parenchyma and Collenchyma are living cells, whereas how can we say they are living? From the diagram we can see that they have nucleus and vacuole, that's why you can say they are living. Sclerenchyma are dead cells, they are empty from inside. Next we come to the cell wall. Parenchyma have very thin cell wall, they have no extra deposit, so the cell wall are basically made up of the basic material which is cellulose. Collenchyma, they are polygonal in shape, and the corners, they are angular at the corners. We can find that the cell wall are thick at the corners, and this thickening at the corner is due to a complex carbohydrate which is called pectin. For Sclerenchyma, the cell walls are thick throughout. So uniformly they are thick, and that thickening is due to another substance which is called lignin. Next we come to the function. So first of all, Parenchyma has a packing function. Packing means similar to thermocol. So when you bring something, so suppose you purchase any gadget, it is packed with thermocol inside the box. So same way, suppose this is the stem and this is the central tissue, the xylem and the phloem. So suppose these are xylem and phloem, they cannot remain hanging in air inside the tissue or inside the stem. They have to be packed, they have to be held in their position so that they don't move from their place. So that packing tissue is parenchyma. Collenchyma is the tissue which provides flexibility to the plant parts. For example, like suppose if I bend this, it goes back. I bend it, it goes back to the original position. So this flexibility to the plant parts, this elasticity which prevents it from breaking, that is provided by collenchyma tissue. Sclerenchyma, it provides protection. Like you find sclerenchyma where in the husk, coconut husk, then the outer covering of nuts and all. So all those are made up of sclerenchyma. So where they are basically located, to know the location, let us have a look at this diagram. This diagram is there in your book. I hope the picture is clear. I'll just increase the brightness. Yeah. So this is a cross section of a stem. This diagram is there in your NCERT book. Now, if you look at one portion, these bundle shaped things that you can see, these are the actually vascular bundle. Vascular bundle consists of xylem and phloem. Now, if you look at one portion, this is one such bundle shown in a enlarged way, and these are the surrounding tissue. So this portion where it is blank, it is actually not blank, it is packed with, or it is, what kind of tissue is present? You can see, you can compare and see the diagram. This portion which is called pith, and there is another portion called the cortex, this area. So the pith area and the cortex area, they are filled with parenchyma. These are packing tissue which will hold these bundles in their position. Next we come to sclerenchyma. There is a region called hypodermis, you can see. So this hypodermal region generally has this collenchyma which provides that flexibility factor. And sclerenchyma are found in older stem and all. So from this you can see the location of the different types of tissue. Okay, so we'll discuss about another layer here, cambium, later on when we'll be discussing about the modifications and epidermis. Fine. So now we see some modifications. We need to know certain modifications. So like for example, in case of certain parenchyma, like the parenchyma found in leaf, suppose I pluck a leaf from this plant. Let's take the whole plant as a whole. So the leaf is green in color and even the stem is green in color. So there are certain parenchyma which contains chlorophyll and they help in your photosynthesis. They can perform photosynthesis. So such kind of parenchyma which contains chlorophyll is called chlorenchyma. Next is erenchyma. Erenchyma are, you have seen a corm, stem. Corm means kosu in Assamese. Or suppose I have taken a banana, a midrib of banana. So if you can see here, if it is getting focused, if you can see, there are lots of empty space in between. So such kind of parenchyma which have lots of air space in between, they are called erenchyma. And why we need erenchyma? Erenchyma is required to provide buoyancy, especially you find them in aquatic plants like in water hyacinth and all. There is one more very important layer which is called epidermis. Derm means cover, epi means on top. So the outermost layer. So any plant part you talk about, whether you talk about leaves, whether you talk about stem and root, all these plant parts, they have an outermost layer, and that outermost layer is called the epidermis. Now how the epidermis is modified in the different parts, like in case of leaf, I'll just show you one more diagram out here. So if you take a cross section of a leaf, cross section of the leaf means if you take the thickness, if you study the thickness of the leaf, so this is the cross section of the leaf. So you see so many cells are present in that thickness of the leaf. So on the top there is a layer of single cell, and this layer of single cell, it is called upper epidermis. And on the lower side of the leaf also there is a single layer cell, single cell layer, and that is called the lower epidermis. So the entire leaf on top and on below, they are covered by this epidermis. Now what happens in upper epidermis? You have a waxy secretion of a substance which is called cuticle. So cuticle, actually this layer cuticle, it is a waxy layer which prevents excessive evaporation and prevents excess loss of water. It is mostly in desert plants you find this cuticle layer very prominent. And lower epidermis, if you take the lower surface of the leaf and see under the microscope, you'll find very fine pores are there. You have learnt in lower class that there are some fine pores called stomata. So those stomata are present. So when we talk about stomata, each stomata is actually the pore, and each stomata is guarded by two bean-shaped cells, and these bean-shaped cells are called the guard cells, and this space in between, that space is actually called stomata. So the lower epidermis of the leaf, it will contain that stomata. Next we come to epidermis of root. So epidermis of root, so root, you know, they have very fine hair-like structure which are called root hairs. So what root hairs are made up of? Let us have a look here. There's another diagram which shows the cross section of a root. So if you take a cross section of a root, you'll find that the epidermis of the root, there the epidermal cells, they have fine extensions, and these fine extension is called the root hair. So what is the purpose of this extension? It increases the surface area for absorption of water and minerals from the soil. So that is the function of the root hair, and that is one modification you can see in epidermis of the root. Now we come to epidermis of stem. So the green young stem, they have normal epidermis, but as the stem becomes older, in case of trees and all, so the epidermis is actually replaced by another layer which is called secondary meristem. Now what is secondary meristem? We need to understand this concept. What I told you that meristematic tissue, when they divide and when the division is over, they stop dividing and they get converted into permanent tissue. Now what happens in some cells, like in old stems, some of these permanent cells will once again become meristematic in nature. They will regain their capacity to divide, and when they regain their capacity to divide, then they are called secondary meristem. So what happens in case of old stem? Suppose there is the cross section of the stem. So I'll show you the diagram that is there in your book, the first diagram. Yeah. So you can see a layer called cambium here. Can you see that? So this layer called cambium and a part of this cortex also gets modified as secondary meristem. So two layers are formed of secondary meristem. One is cambium layer becomes secondary meristem, and a part of that cortex layer becomes secondary meristem. Now when there is formation of secondary meristem in this portion just below the epidermis, then they will keep on growing, dividing, because they are meristematic now, they will divide. They will form a group of very closed packed cells, and they will have lots of waxy deposit of a substance called suberin. So suberin is a complex waxy chemical. So they will have deposit of suberin, and they will make that layer, the suberin will make the layer waterproof and insect proof. So it provides or helps in formation of that protective layer, and that protective layer we commonly call as bark and cork. So bark is more harder, cork is a softer layer, but they are made up of that secondary meristem. So that is a kind of modification we can see in the epidermis of the stem. Next we come to complex permanent tissue. So when we are talking about complex permanent tissue, they are of two types, xylem and phloem. Together xylem and phloem is called vascular bundle. These are conducting tissue. Just like we have blood vessels for the blood to flow, similarly in plants they have xylem and phloem. So what are the components? Why complex? I told you before, because they are made up of different types of cells. And what are the different types of cells? There will be xylem parenchyma, xylem fibers, tracheids and vessels. For xylem, phloem are made up of phloem fibers, phloem parenchyma, sieve tube cells and companion cells. Now in xylem, these three, xylem fibers, tracheids and vessels, are dead cells. Xylem parenchyma is living cell. In phloem, phloem fiber is dead cell. Phloem parenchyma, sieve tube cell and companion cells are living cell. Now, the function of xylem, it is to carry water and minerals from roots to the leaves, and that function is performed by tracheids and vessels. So these are the functional component. Parenchyma provides acts as a packing tissue in between all these different types of cells. Fibers provide mechanical strength. And similarly here also parenchyma will act as packing tissue and fiber will provide mechanical strength. The function of carrying water and minerals will be performed by tracheids and vessels. What is the function of phloem? Phloem carries food and other substances from one part of the plant to other part. But it is mostly from the leaves to the roots and the other parts. And that is done by sieve tube cells assisted by companion cells. So these two are the functional cells. So how vessels looks like? Actually they are made up of cylindrical cells, and the cell wall in between that gets in due course of time that gets broken out, or and all the cells, they form continuous tubular structure, fine tube like structure. Those are called vessels. So through this, and tracheids are more or less spindle kind of cells, but they do have in their cell walls there are fine pores, and through those pores they maintain the continuity which helps the water to move forward. So that's vessels and tracheids. Now we come to phloem. So phloem, you see sieve tube cells, through these the food will flow. Now why it is called sieve tube? Because the cell wall between the two cells, they have fine pores just like that of a sieve. I mean je saloni use koru, the sieve that we use, so it looks like that sieve, that's why it is called sieve tube cell. Now through this food is flowing. So, and this is a living cell. So since it is living, it should have cell organelle, nucleus, vacuoles, everything. So it actually does not have all this. Why it does not have all this? Because if it has vacuole and nucleus and cell organelles, it will hamper the flow of food. So that's why this cell is relatively empty. It has only cytoplasm. Is that clear? Now the question is, if it is a living cell and it does not have the cell organelle, then how it remains living? So the answer is, all the function of the sieve tube cell is carried out by another cell which is located besides and has a prominent nucleus, that is called the companion cell. That's why the name companion. Companion means what? Friend. So these companion cells help in function of the sieve tube cells. And why sieve tube cells are empty? Because they have to help in flow of food. Now one more question. Okay, they need to help in flow of food. So if they would have been dead, then it would have been better. Dead cell means empty cell, so the food could have moved easily. So why the cells are living? That is another question. So the answer is, why the sieve tube cells are living? Because you see, the food that is flowing through the phloem, it needs to be controlled, because every part of the plant will not require the same amount of food. For example, this plant. Now this is a big mature leaf, it will not require so much of food right now, but these are young growing leaves, so they will be requiring more food. So the food which will be manufactured in the leaves, more food needs to be sent to these parts compared to these parts. Is that clear? So for regulation, something which is dead cannot regulate. So if this is a dead cell, it cannot regulate. So for that regulation, this cell is living in nature. Okay, that is one concept. And why these are dead? Because they don't have to control. Water enters from the soil through the root hair and it will reach to the xylem. And how the water moves out through the xylem? It is very simple process. This xylem is present till the tip of the finest vein of the leaves. Even the finest vein has xylem and phloem. So what happens during daytime? Water will move out as water vapor by the process of transpiration. We know that loss of water as water vapor from the aerial parts of the plant is called transpiration. So when transpiration takes place, there will be a kind of vacuum which will be created in the xylem, and that vacuum will pull the entire column of water in the upward direction. And why transpiration is occurring? It is due to heat of the sun. So the cells need not control that flow of water through the xylem tissue. That's why these are dead cells. So overall, that's the concept and the details of simple permanent tissue and complex permanent tissue. Thank you. In the next video, I will be discussing about animal tissues.